Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this video, I will be explaining you an introduction to proteins, and then we will classify proteins, and also we will learn higher order of protein structure. Let's get into the details. Now, proteins are nitrogenous macromolecules which are composed of amino acids. Basically, amino acids are the building blocks of proteins. So that's why proteins are called as polypeptide chains, because one amino acid is joined with another amino acid by a peptide bond. That's why it is called as polypeptide chain. Okay. Now, as you know, amino acid has got alpha carbon in the center. We have seen that in our previous lecture. And it has got a primary carboxyl group. It has got a primary amino group. It has got H and the side chain. Okay. Now, that means an amino acid has got carbon, it has got oxygen, it has got hydrogen, and also it has got nitrogen. So there is nitrogen in an amino acid. So proteins are made from amino acids. So that is why it is referred as a nitrogenous macromolecule. In fact, 16% of a protein molecule is composed of nitrogen. So this is a major nitrogen containing macromolecule that we have and we get into our body. Because when we take proteins, proteins are the source of nitrogen for us. So remember that point. Now, with that introduction, let's classify proteins. Now, proteins are classified based on their shape and size. Example, we have fibrous proteins. Example, for fibrous protein, we have collagen and keratin. And then we have globular proteins. As the name says, they are globular in shape. So, example for globular protein, we have hemoglobin, myoglobin, a lot of enzymes that are there in our cytoplasm and in most other subcellular organelles. They are globular proteins. And then we have proteins can be classified based on their functional properties. That can be defense proteins. Example, we have immunoglobulins. Then we have contractile proteins, actin, myosin. These are contractile proteins. Proteins can be classified based on their nutritional value. And that can be a complete protein and incomplete protein. So complete proteins, example, we have casein in the milk, and they contain all essential amino acids that we have seen in the previous lecture. Incomplete proteins like pulses which lacks methionine, or cereals which lacks lysine. So there is incompleteness there because they don't contain all essential amino acids. Now, on the based on the solubility and physical properties, proteins can be classified into simple proteins. Simple proteins, they can be hydrolyzed, and upon hydrolysis they will give all amino acids. Or they can we can classify proteins as conjugated proteins. Conjugated proteins are the ones which will give, along with the amino, when they are hydrolyzed, along with the amino acid, some other component will be released from a conjugated protein. Example, say glycoproteins. It is composed of carbohydrate and amino acids. Then we have mucoproteins, chromoproteins, nucleoproteins. So protein plus a prosthetic group containing molecules are referred as conjugated proteins. So some more examples here, going with the previous categorization, like based on the functional properties. So we have respiratory proteins, hemoglobin, cytochromes, myoglobin. Then structural proteins, collagen, keratin, enzymes, glucokinase. Example given here, hormones, we have thyroid hormone. These are all like based on the special properties we have classified protein. And based on the nutritional value from this particular categorization, so we have seen from the previous slide. So we have complete protein, incomplete protein, and then we have poor protein. Poor protein, a gene from corn, which is deficient in many essential amino acids there. And then from this category, classification of proteins based on solubility and physical properties. We have simple proteins, conjugated proteins, and also we have derived proteins. So derived proteins, basically these are the proteins which are derived from primary proteins. Like example, when we cooked meat, secondary derived proteins are like proteases, peptones, peptides. These are actually derived from a primary protein molecule. So that's a simple classification of proteins based on the shape and size, functional properties, nutritional value, and solubility and physical properties. All right. Now, let's look into a little bit of hydrolysis process, protein hydrolysis. So, hydrolysis it's a complete hydrolytic decomposition of a protein. When you break a protein, when you add water and break a polypeptide chain, when you do the hydrolysis process. So, this is the stages of hydrolysis that is given here. A protein is initially hydrolyzed into protein. Protein further hydrolyzed into metaprotein. Metaprotein into protease. Protease into peptones. Peptones into peptides, and peptides into amino acids. So, this is the order of hydrolysis that you may be required to remember. That is proteins to protein, metaprotein, protease, peptone, peptides, and amino acids. Note that the products from protein to peptone up to peptone. So, protein to peptone, that is the one which will give a positive biuret reaction. So, that means in this particular order, so we got to go all the way until protein to peptone. So, all these molecules will give a biuret test positive biuret reaction, and are relatively, these are large molecules. And then peptides and amino acids, they don't give a positive biuret test. So, they are abiuret, means they don't answer for biuret test, which is a test for proteins and up to peptones, not for peptides and individual amino acids. Remember that. All right. Coming to biological functions of our body proteins. So, we will take a specific example and understand what is the biological functions of each of these proteins. Like enzymes. Enzymes are biological catalysts, as you all know. They enhance the rate of reaction. Some of the names of enzymes that is given in the table here, of course, like there are hundreds of enzymes that you are going to study in the metabolism. So, example amylase, ribonuclease, trypsin, these are digestive enzymes. And then we have enzymes in metabolism like glucokinase, hexokinase, pyruvate kinase. A lot of enzymes are there. So, basically they are all biological catalysts. Remember that. And then the structural proteins. Structural proteins, example, we have collagen, elastin, keratin, fibrin. These are all structural proteins which present in the extracellular matrix and give structure to a tissue. Okay. And then the transport proteins, as the name says, they transport something like hemoglobin. It transports oxygen. Myoglobin, it transports oxygen again, but it's a storage form of protein. Basically, it stores oxygen in the tissue. And then lipoproteins, it carries lipids from one place to another place. Serum albumin, membrane transporters, they are all like transport proteins. Then we have hormonal proteins like insulin, growth hormone, and then we have corticotropin, then thyroid stimulating hormone. These are hormones which are protein in nature. And then regulatory proteins, we have repressors, inducers, transcriptional factors. These are all regulatory proteins. And the contractile or motile proteins, we have actin, myosin, tubulin in the contraction process. And continue with that, we have like nutrient and storage proteins. Example, we have gliadin in the wheat, ovalbumin, casein, which are all nutrient proteins. Defense proteins, as you all might be knowing, it is antibodies. Immunoglobulins are defense proteins. And then the blood proteins, we have fibrinogen, thrombin, which is participating in the clotting process. Toxin proteins, we have snake venom. Snake venom contains some of the proteins within it. And then vision protein, we have rhodopsin. Walt's visual cycle, we are going to study that in vitamins. Rhodopsin is it's actually chromoprotein. It has opsin plus 11-cis retinoldehyde. Okay. Coming to proteins higher order structure. Now, the proteins can be classified based on their structural organization into primary, secondary, tertiary, and quaternary. Okay, so primary structure, it is nothing but it is a sequence of amino acids joined with one another by a peptide bond. So simple primary structure I am writing here. So it is amino acid 1, amino acid 2, amino acid 3, amino acid 4, 5. So all these amino acids are joined with one another by a peptide bond. So that type of arrangement is called as primary structure. Now I'll just write it as a linear chain like this for simplicity. This is the primary structure. Okay. Now if this folds itself and gets some shape like this, and this is a secondary structure, that means an interaction within the polypeptide chain which will give a certain shape to a protein molecule, we call it as a secondary structure. Now this secondary structure protein molecule which has got amino acids with their side chains, that is R groups present in each one of these amino acids, these R groups can interact with one another depending on their characteristics, okay, and that will give a certain shape to a protein molecule, okay, and that shape we call that as a tertiary structure of a protein molecule, and one tertiary structure we call it as a domain or a subunit, one domain or one subunit. That is a tertiary structure of a protein molecule. And then if a protein has got more than one tertiary structure, something like this, and they all have R groups, some of their R groups which are free in each of these domains. I am just writing R here. And these R groups, they can interact with one another. So R group of a domain number 1, R group of domain number 2, if they interact with one another with their depending on their side chain characteristics, and that entire structure now is referred as a quaternary structure. So this is how a higher order protein structures classification is done. So primary, secondary, tertiary and the quaternary structure. Remember that. Now let's get into primary structure little more in detail than what I have explained. So as I said before, primary structure, it is a sequence of amino acids joined with one another by a peptide bond, and this sequence, it is genetically determined. So that means, as it is shown in the figure here, we have alanine, cysteine and valine. 3 amino acids are there in a short peptide here. So first amino acid is alanine, second amino acid is cysteine going from left to right, and third amino acid is valine. Always remember, when we write an amino acid, we always start from the amino group and go all the way towards the carboxyl group. So amino end, that is the N terminus, and this is the C terminus. So we go from N terminus to C terminus. So first amino acid here is alanine, but it is part of a polypeptide chain. That's why I always remember, we don't write it as alanine, we write it as alanyl. So alanyl, remember that. So we end it as YL. And the second amino acid shown here is cysteine, but we don't write it as cysteine, we write it as cysteanyl. We end it as YL. And the last amino acid, which is not attached further with any other amino acid, we just use the name valine here. So any amino acid in this place, we just write it as it is. We don't add YL to the last amino acid. Always remember, that's how we write a peptide bond. When we show a specific amino acid in a peptide bond, we write it like that, starting as, means ending as YL for each amino acid, but the last amino acid, we don't add YL there. So that is what you can see here in the figure. Now, the alanine here is interacting with the cysteine by a peptide bond. This is the one that is shaded. As you see here, carbonyl oxygen of primary carboxyl group of alanine is going to interact with nitrogen hydrogen with the primary amino group of cysteine. So it is CONH bond there. That is the peptide bond. Peptide bond. And peptide bond is a strong bond, covalent in nature. Remember that. Okay. So that is the bond which is responsible for a primary structure of a protein molecule. So let's look into that peptide bond a little more. So peptide bond exhibit partial double bond characteristics, and it is in trans configuration. As you see, this is in trans configuration, coplanar and semi-rigid. These are some of the characteristic of a peptide bond. It is a covalent in nature. Covalent bonds are strong bonds, and then it has got a partial double bond characteristics, trans configuration, and it is coplanar and semi-rigid. Okay. Now, so any alteration in the amino acid sequence. So as I have written, okay, in amino acid 1, amino acid 2, and then amino acid 3, amino acid 4, amino acid 5. So I'll end it as O minus here, NH3 plus on the left hand side. Okay, so if my first amino acid was alanine, second amino acid is say cysteine, third amino acid is valine, fourth amino acid is tyrosine, fifth amino acid is methionine. So this is a sequence of amino acids determined by a genetic sequence. Okay, now what if that sequence changes? Okay, so if a genetic codon, which is a triplet 3-letter codon, what if it changes because of mutation in a gene coding for that particular peptide bond? So that means it is going to change an amino acid. Example, if there is a mutation in a codon coding for cysteine, instead of cysteine, if it codes for say glutamate or something, or instead of say glutamate, if it is replaced by valine or something, if there is any change in the sequence of amino acid because of mutation, it will alter a total function of a protein molecule. It will alter the charge, it will alter the function of a protein molecule, and that may give rise to a disease condition. That's what I have written here. So alteration in the sequence of amino acid from a original amino acid, so it may lead to disease. Example for that, we have sickle cell anemia. Sickle cell anemia, especially in the beta chain of hemoglobin, where glutamate at the 6th position will be changed with valine, hydrophobic amino acid valine at the 6th position of glutamate. It will be changed, and that may lead to sickle cell anemia. Okay, especially if it happens in 2 of the beta chains in red blood cells. Okay, coming to something called as atypical peptide bond, which is also referred as pseudo peptide bond or isopeptide bond. Remember, atypical peptide bond is generally formed between an amide, it's an amide bond formed between an amino group and a carboxyl group of at least one of which is not on the alpha chain. It will be like this. So we have a alpha carbon. So we have COO minus, NH3 plus, and then we have H and R. Okay. Now, if this particular carboxyl group, instead of interacting with another amino group attached to the alpha carbon, so this is the alpha carbon, COO minus, H and R. If these two interact, that's a normal peptide bond. Okay. Now, instead of that, if I write something like this, so alpha carbon, H, amino group, primary carboxyl group, and then in the side chain I will write like this, CH2, and then CH, C double bond O, and NH2, and this NH2, if it interacts with this carboxyl group, so side chain NH2, that is a amide nitrogen. If amide nitrogen is interacting with the primary carboxyl group of any other amino acid, still that makes a kind of peptide bond. It will be CONH, but it's not a peptide bond. It is referred as pseudopeptide or atypical peptide bond. So sometimes in a polypeptide chain, this type of peptide bond formation can go on. So which are referred as isopeptide bond. That's what is shown here. It is between lysine and asparagine, where isopeptide bond is aspartate or asparagine, isopeptide bond is possible there. Okay, so remember that point. Coming to some of the characteristics of atypical peptide bond or isopeptide bond. So isopeptide bonds generally happens post-translationally, means once the protein synthesis is done and later this kind of interaction can go on. They can be found spontaneously or enzymatically. They can produce a stable linked protein dimers, multimers, complexes. They make proteins resistant to like proteases that cannot be hydrolyzed, And cyclic peptide antibiotics like tyrosine, gramicidin, they contain atypical peptide bond. Remember that. Some of the antibiotics like tyrosine and gramicidin, they are example for molecules that contain isopeptide bond. And heptapeptides like dermorphin, deltorphin, so they are also containing atypical peptide bond. So on the applications here, they can be used to develop a new peptide tag called isopeptag. It is used in vivo protein targeting, fluorescent microscope imaging techniques. So these are some of the uses of isopeptide bonds there. Okay, now let's look at some of the biologically important peptides. Since we are in peptide bond like primary structure, so primary structure, one amino acid joined with another amino acid making a dipeptide, so two amino acids together, which is a dipeptide. So although it kind of misnomer here, di means two, peptide bond, but actually dipeptide contains two amino acid but one peptide bond. Remember that. So example, we have carnosine and anserin. These are the peptides found in muscle and they activate myosin ATPase enzyme. Then the tripeptide example, we have glutathione, GSH. Glutathione is gamma glutamyl cystenyl glycine. Gamma glutamyl cystenyl glycine is glutathione, which is in reduced form it is called a GSH. It is an important intracellular antioxidant. Remember that. Tripeptide, other example we have like thyrotropin releasing hormone, which is secreted by hypothalamus and helps in release of thyroid stimulating hormone. Pentapeptide, we have enkephalin, which is found in brain and it inhibits the sense of pain. Some more examples here, we have octapeptide containing 8 amino acids. Angiotensin 2 is an example for octapeptide. Nonapeptide, vasopressin is an example for nonapeptide secreted by posterior pituitary gland and also it regulates water in our body. And nonapeptide another like bradykinin, which is a vasodilator peptide. So these are some of the examples for important peptides that we have. Coming to secondary structure of a protein molecule. Now secondary structure is due to it adopting an amino acyl residues that adopt a pi and psi angles and that will give rise to specific shape to a protein molecule. Basically a polypeptide chain which will adopt an amino acyl group residues where pi and psi angles are made. Okay, so two most common types of secondary structures we have is alpha helix and beta sheet. And note that when you draw a Ramachandran plot, so it will be something like this. Okay, so in a Ramachandran plot, alpha helix and beta sheet, they will be falling on the left side, both the lower quadrant and also upper quadrant. So upper and lower left quadrant, that's where you are going to find alpha helix and beta sheet. Within that, as you see here, they fall within the lower and upper left quadrant of Ramachandran plot respectively. That means alpha is at the lower left and beta is on the upper left quadrant of Ramachandran plot. Remember that. Now, whether it is an alpha helix or beta sheet, so they both are formed by hydrogen bonding between the atoms of the peptide bond. Remember that. So, formation of alpha helix or the beta sheet, it is all because of the hydrogen bonds that are made between the peptide bonds. That is, peptide bond is CONH. We have carbonyl oxygen, nitrogen, hydrogen. So, this carbonyl oxygen, it can interact with the nitrogen, hydrogen of another peptide bond. Not the same peptide bond, a neighboring peptide bond. There can be interaction between carbonyl oxygen and nitrogen, hydrogen. Similarly, nitrogen, hydrogen of one peptide bond can interact with carbonyl oxygen of another peptide bond. So, that kind of interaction which will adopt a pi and psi angles, right? That will give rise to secondary structure to a protein molecule. Let's look into one of the secondary structure which is found at the lower left quadrant of the Ramachandran plot, and that is alpha helix. Now the alpha helix, it is a regular right-handed coiling of a polypeptide chain. Okay, so as you see, if you look at, if you hold the thumb like right hand and the thumb which is facing on the right side, okay, this is a right-handed coiling. That is how the alpha helix look. It's a regular right-handed coiling. Look at this. It goes like this and then turns and go this way. So hydrogen bonds are formed between the peptide bonds to make alpha helix. So hydrogen bonds are the ones that are responsible for the formation of alpha helix. How exactly it is done? So formation of hydrogen bonds within the polypeptide chain that gives alpha helix, it follows a rule called N+4 rule. N+4 rule. What is N+4 rule? N+4 rule is a peptide bond, one peptide bond. Say if you say it as, say it will be like this, amino acid 1, and then I will write amino acid 2, amino acid 3, amino acid 4, and then amino acid 5, amino acid 6, like that, and then I'll write it as CO minus, and this is NH3 plus. Okay, now this is the first peptide bond. I'm writing it as CONH, first peptide bond, and now this first peptide bond, it is going to make a hydrogen bond with the peptide bond which is 4 amino acid ahead of it. That means this is N, you write it as N, then number 1, 2, 3 and 4. So this is fourth peptide bond, CONH. So carbonyl oxygen of this peptide bond, it can interact with nitrogen of the fourth peptide bond which is ahead of it. Okay, so or it can be behind it also if there were any peptide bond. So like this, N+4 rule, based on the N+4 rules, there can be numerous hydrogen bond formation between the peptide bonds, and that will give rise to a certain shape to a polypeptide chain, and whatever the shape that it assumes, that is what is referred as alpha helix of a protein molecule. Remember that. So alpha helix formation is because of hydrogen bond formation, and the rule that it follows is N+4 rule. Okay, that means nth peptide bond, it is going to interact with the make hydrogen bond with the fourth peptide bond which is ahead of it or behind it. Now let's see how many amino acids are there in one turn of an alpha helix. That information is given here. As you see, alpha helix has 3.6 amino acids per turn. Okay, so in this one turn here, if we go from, which is basically referred as one pitch, so if we go from here to here, alright, so it has 3.6 residues. That means 3.6 amino acid in that, and also the distance between that entire length of that one turn is 0.54 nanometer, and also the distance between two atoms present in that is 0.15 there. And also see the number of atoms present in that one turn is 13. There are 13 atoms present in one turn of an alpha helix, and that's why it is called as 3.613 helix. Now, one of the amino acid that you must remember in alpha helix is, it is the presence of proline or hydroxyproline. Whenever proline comes in or hydroxyproline comes in, the alpha helix do not continue, because proline and hydroxyproline do not allow alpha helix to continue, that smooth alpha helix to continue there. So it basically alpha helix ended there in a polypeptide chain whenever proline comes in or hydroxyproline comes in. That's why it is called as helix breaker. Proline or hydroxyproline is simply referred as helix breaker. Remember that. Now let's look at some of the amino acids and their relation with the alpha helix formation. Now amino acids like alanine, glutamate, methionine, leucine, they all tend to be present in alpha helix. These are some of the common amino acids that you should look for in an alpha helix of a polypeptide chain. It is alanine, glutamate, methionine and leucine. And the most abundant amino acid out of all present in alpha helix is methionine. Methionine is the most abundant amino acid present in an alpha helix, and that is followed by glutamate. The frequency of appearance of methionine to glutamate, it is written here. It is 1.47 relative frequency for methionine. 1.44 is the relative frequency for glutamate. And also amino acid that is least present in alpha helix is proline, because in fact proline is referred as helix breaker, proline or hydroxyproline. So remember that. So it is the least amino acid that is present in the alpha helix. In fact, that breaks alpha helix. Now, amino acids that do not favor alpha helix, let's look into that. So amino acids with the branches at alpha carbon atom do not favor alpha helix formation. It's like valine, threonine, isoleucine. So they all have that branches there in the attached to the alpha carbon. So should not be part. Basically, they will disrupt the stability of alpha helix. And other amino acids that disrupt the stability are like serine, hydroxyl containing amino acid serine, aspartate and asparagine. Now, proline also disrupts the stability of alpha helix. In fact, it breaks the alpha helix. And whenever glycine comes in, usually glycine comes in along with the proline. So glycine induces bends in the alpha helix. So remember these points related to the role of specific amino acids with alpha helix. Now coming to beta sheet. Beta sheet, it's a zigzag or pleated pattern of a secondary structure of a protein molecule. Just like alpha helix, in the beta sheets, there can be R groups which are adjacent to each other and which will project in the opposite direction present in the beta sheet. It will be like this. So basically beta sheet formation needs two individual polypeptide chain which are running either in the same direction or opposite direction. If it is running in the same direction, we call it as a parallel beta sheet. If they run in opposite direction, we call them as a anti-parallel beta sheet. Okay. So the bond formation between these two peptides to make beta sheet is again between the peptide bonds CONH. Just like alpha helix, it is the interaction between two peptide bonds. So one carbonyl oxygen of one peptide bond interacts with nitrogen and similar to that. So there will be cross interaction like this between two polypeptide chain and they come kind of closer, and that's why they get something like pleated appearance. And the R groups, either they will be above the plane of the ring or below the plane of the ring. So this is how a beta sheet formation is done. So beta sheet is formed by hydrogen bonding between peptide bonds between two polypeptide chain, or it can be a single polypeptide chain which is bent over because of proline and glycine, which will lead to, it will induce bends when it is bent over, and there can be interaction between this polypeptide chain which is sufficiently long and it is bent over. So that can also give rise to beta sheet. Remember that. So now this beta sheet is rich in hydrophobic amino acid. Always remember, whenever a protein has got an hydrophobic core or hydrophobic region, usually that hydrophobic region is the beta sheet. So beta sheets are rich in hydrophobic amino acid, and within that hydrophobic amino acids, valine and isoleucine, they tend to be present in beta sheet. Make sure you remember this point, very important. First point is beta sheets are usually rich in hydrophobic amino acids. Which is the most important amino acids within that hydrophobic category? It is the valine and isoleucine, and within the valine and isoleucine, it is the valine that is most abundant amino acid in the beta sheet. So these are some of the important points that you must be aware of. So hydrophobic amino acid, valine and isoleucine, and within that it is the valine that is the most abundant. And amino acids that are least present in the beta sheet, again it is a proline. Now beta sheets, sometimes they are referred as beta barrels, which will form core of many globular proteins. Now let's move on to see some of the atypical secondary structure which are non-regular, non-repetitive kind of structure, because alpha helix and beta sheets are regular and repetitive structure, whereas loops, turns, bends and coils, these are non-regular, non-repetitive structure which will come once in a while, and that will actually give rise to a particular shape to a protein molecule. So, example, in a globular protein, where a single polypeptide chain, it has to bend over and give that globular shape. So, in order to achieve that, there will be a lot of bends, turns, loops, coils need to be going on along with alpha helix and beta sheets. That's how you get a long linear chain folded into a ball-like thing. So, obviously, there is a role of loops, turns, bends and coils there. A beta turn involves 4 amino acid residues, and within those 4 amino acid residues, as I have shown here, so 1, 2, 3 and 4 here, within that proline and glycine often they are present in beta turns, loops, turns and coils. Remember that. Now, loops, they will bridge domains that are responsible for binding of substrates. Often they contain amino acid residues that will participate in catalysis process. Like in an enzyme, so there is an active site in an enzyme, and in that active site, the loop, basically it has got a specific type of amino acids. Many loops and bends, generally they present over surface of proteins, constitute readily accessible sites or epitopes for recognition or binding of antibodies. And most abundant amino acid in beta, bends, loops and turns is proline, and the proline is followed by glycine. So that means within the turn, because out of those 4 amino acids that I said, if turn is like this, so amino acid 1, amino acid 2, amino acid 3 and amino acid 4, within that proline is the most frequent one, that is frequency of occurrence is 1.91, followed by glycine. That means in beta turn, loop, turns, coils and all that, so you are going to expect proline most frequently and followed by glycine. Always remember, proline usually it follows glycine. Alright, so that's the beta turns. Now let's look at tertiary structure. Tertiary structures, they are interaction between alpha helix, beta sheets or beta bends. So especially R groups which are free so far, because in alpha helix it is interaction between the peptide bond, in beta sheet it is interaction between the peptide bond, hydrogen bonds are formed, and that's why you get like alpha helix or beta sheet. R groups interaction we have not seen yet. So R groups present in each of these amino acids, they can interact with another R group depending on the side chain characteristics. Like if we have a carboxyl group COO minus in the side chain, and then we have NH3 plus, so they can make a salt bridge formation, ionic interaction like that. So depending on the side chain characteristics, there can be formation of different types of interaction between the R groups, and that will give rise to a shape to a protein molecule, and that is a 3D shape. We call it as a domain or a subunit, and that's the tertiary structure to a protein molecule. Now, hydrogen bonds, hydrophobic interactions, ionic bonds, disulfide bridges, weak van der Waals forces, all of them make tertiary structure, because all of this kind of bonds are possible between R group interaction. So simple proteins will have a single domain, and complex proteins will have more than one domain or one subunit. So if it is a simple protein, it just like have one tertiary structure, one subunit will be there. If it is a complex protein like say hemoglobin, hemoglobin has got 4 subunits, so that means it has 4 tertiary structure. Now within that tertiary structure, especially say if it is a oxidoreductase class of enzyme, so the domain present in that oxidoreductase class of enzyme where NADP, because or NADs or NADP, because oxidoreductases, what they will do, when the substrate is oxidized to product, NAD is reduced into NADH plus H plus. That's a reduction. So all this kind of action will be going on in a special fold in these oxidoreductases called Roseman fold. Remember that. Okay, so higher order protein structures are stabilized primarily and often exclusively by non-covalent interaction. That is, tertiary structure which will make different kinds of bonding, which will stabilize all these tertiary structure, like we have hydrogen bonds, hydrophobic interaction, ionic bonds, and then disulfide bridges, weak van der Waals forces. Out of all these non-covalent interaction, the most predominant, and these are the ones that will stabilize tertiary structure and also quaternary structure. I am going to explain you in the next slide. So out of all this, the most important one, most important non-covalent interaction that will stabilize this tertiary structure is a hydrophobic interaction. Remember that. Hydrophobic interaction is the principle among all non-covalent interaction that will stabilize tertiary structure of a protein molecule. That's point to be remembered. Now, coming to quaternary structure. A protein that has got more than one subunit. Here, so we have alpha 1, alpha 2. I am just naming it as alpha and beta. Beta 1 and beta 2. And then we have a heme group there. So basically it is a hemoglobin or something. So 4 subunits are there. All these 4 subunits, they are interacting with one another. Basically the R groups in one subunit interact with R group in another subunit, and then R group. So like this R group interaction between these subunits, that overall it will give a shape to a protein molecule, and that shape we call it as a quaternary structure. Remember. Now, what are the bonds that are responsible for quaternary structure, and what type of quaternary structure proteins we have? What if there is a similar subunit? What if there is a different subunit? So let's look into little more details on that. Now the interaction between domains or subunit will make this quaternary structure. If all the subunits are of same type, like if all of this, if there are 4 subunits and all those 4 subunits are of alpha type, alpha 4, that we call it as a homotetramer, because all of them are the same. Or if it is like 2 alpha and 2 beta, just like in adult hemoglobin, so we have 2 alpha and 2 beta, so it is referred as heterotetramer. This is how the naming goes. Okay, so all these subunits, they are interacting with one another by non-covalent interaction. So the predominant interaction that brings all these subunits together is non-covalent in nature. That is hydrogen bond formation, hydrophobic bond formation, ionic interaction, weak van der Waals forces, disulfide linkages. All of them, they come together and basically make a quaternary structure of a protein molecule. So, and any protein that has got a quaternary structure, it has got higher stability and also higher efficiency. Remember that. And also out of all these non-covalent interactions that are holding on to these subunits together to give a quaternary structure, the most important or the principal non-covalent interaction among all this is hydrophobic interaction. Remember that. Now, how do you determine these protein structure? We have seen like primary structure, secondary structure, tertiary structure and quaternary structure. How do you determine that? So, it can be done by through the 3 mechanisms. Number 1 is X-ray crystallography, where Fourier synthesis process is done, and that will determine the structure of a protein molecule. And then we have nuclear magnetic resonance spectroscopy, NMR spectroscopy. Basically, it analyzes proteins in aqueous solution, because X-ray crystallography, it is analyzing the structure in crystals, because it is there in the name, crystallography, crystals, that is a solid structure. Whereas NMR spectroscopy, it will analyze protein structure when it is in the solution. So it renders possible real-time observation of the changes in conformation that accompany ligand binding or catalysis. That means in NMR spectroscopy, you can also see the catalytic mechanism of an enzyme where substrate is converted to product. That kind of dynamic changes you can measure in NMR spectroscopy. Now, the third technique in determination of protein structure is cryo-electron microscopy. So, it is well suited for detecting conformational states and complexes, analysis of individual components within the heterogeneous samples. That means when the sample contains more than one kind of molecule, like complex molecules, within that complex molecule, you are able to analyze a protein structure. That's one of the advantages of cryo-electron microscope. So these are all the techniques how we can determine protein structure. So in this lecture we have basically classified proteins, and also we have studied organization of proteins, higher order of protein structure. We looked into details on primary, secondary, tertiary and quaternary structure. Thank you.